Bismillahirrahmanirrahim. You might think that the jellyfish and this cheetah over here really have nothing in common, but they do. They both belong to the animal kingdom. And one of the most remarkable phenomena about the animal kingdom is that the early embryonic development of all of the living things that belong to the animal kingdom is the same. In other words, when this jellyfish and this cheetah were zygotes, and then they started to develop, their early development process was very, very similar. And understanding of this early embryonic development, that is, the development of the zygote in its formative early stages, helps us understand the complex anatomy of, for example, the human being. As complex as the human body is, believe it or not, it is quite comprehensible if you understand the embryonic origins of each organ systems. That is what we shall try to understand today. That is, how did this body come about from the earliest stages of the zygote? Well, almost. The embryologic development of mammals or humans is slightly different than what we shall talk about today. But even then, in the very early stages, it is the same. As you know, one of the key features of the animal kingdoms is that the organisms are made of tissues. And you will see that the majority of the animal kingdom is made of three types of tissues. The outermost layer is called the ectoderm, the middle layer is called the mesoderm, and the innermost layer called the endoderm. So these three layers of tissue give rise to all the different types of tissues and organ systems in an organism. In human embryology, for example, the ectoderm layer gives rise to the skin and the brain and spinal cord, whereas the endoderm gives rise to the lining of the gastrointestinal tract. What we shall learn today is how a zygote evolves and develops into an embryo that has these three layers. The most primitive of the animals of the animal kingdom only have two layers. A zygote, as you know, is a diploid cell, and it's the result of the joining of the sperm and the egg. Once a zygote divides, it's no longer called a zygote, it's called an embryo. That is, after the first cell division, where one cell gives rise to two cells like this, the zygote is no longer called a zygote, it's called an embryo. And hence the term embryonic development, because from here on we shall be talking about the development of the embryo. So here the zygote first divides into two cells, and then they divide again to give 4 cells, and from 4 to 8, and then 8 to 16, etc. Now in the early stages, the cells divide, but they do not grow. So the size of the embryo does not really change, as you can see over here. This process of cell division without cell growth is called cleavage. Now, once it forms a ball of cells like this is called a blastula. Now, these cells started out as a ball of cells, but then the cells start secreting fluid inside like this, and they create a space that's fluid-filled. This stage is called a blastula. In the next stage, one part of the ball starts to dimple in. You can just imagine somebody pushing a finger in like this, making a dimple in this ball of cells. At this stage, the embryo is known as the gastrula. And this opening right here is the beginning of the formation of the intestinal tract or the gastrointestinal tract or the digestive tract of the living thing. Because as you know, one of the cardinal features, one of the most important features of the animal kingdom is that they are heterotrophs. That is, they gather their energy from other living things. So they must eat, and if they eat, they must digest. Thus, one of the earliest developmental features of the animal kingdom is the formation of the gastrointestinal tract, so to speak. Okay, so here you see early cellular division without the change in size. Now this shows you the blastula, the fluid-filled ball of cells. Now this is a nice animation of the development of the blastula and the gastrula, although it's in a different language. You can see the invagination of the tissue giving rise to the 2 layers of cells and the formation of the blastopore. Okay, so let's label some of this. This opening here is called a blastopore, and now you can see we have 2 layers of cells. This outer layer cell is called the ectoderm, and the inner layer cell here is called the endoderm, and this hollow area over here, this is in fact the primitive gut. Now some living things are really only 2 layers thick, like this. And this in fact is the basic shape of jellyfish. It has an ectoderm layer, which is blue over here, and this endoderm layer, which is yellow in this diagram. As beautiful as a jellyfish is, it's really only made of 2 layers of tissues. The dermal cells, which gives rise to the epidermis, and the endodermal cells, which gives rise to the gastric sac or the gastric cavity, where digestion occurs. Well, we had said there were 3 layers. Where does the 3rd layer come from? Well, the 3rd layer comes from the endoderm. Some of the cells from here break off and form a massive tissue like this. Sometimes they come from this area, sometimes they come from this area, depending on which species we're talking about. So a 3rd layer of tissue is thus generated. And this, of course, is the mesoderm, the middle layer. So now we have 3 layers: the mesoderm, the middle layer, the ectoderm, the outer layer, and of course the endoderm, the inner layer. Ectoderm, mesoderm, and endoderm. Now the mesoderm is a very creative layer. It can do one of several things. One of the things that it can do is like this. It can basically go on to obliterate this other space that was over there in the beginning, creating a fluid-filled cavity of its own like this. Now, as you can see, this living organism has 3 layers. Here's the ectoderm, mesoderm, and of course the endoderm. Now mind you, except for the sponges, which is the most primitive of all animals, all animals in the animal kingdoms, they have tissue differentiation. They have either 2 layers of tissues or 3 layers of tissues, ectoderm, mesoderm, and endoderm, and these give rise to all different organs of their bodies. There's no 4th layer. That's it. And if you think about it, that is quite remarkable. Now in this picture over here, something remarkable has also happened. This blastopore, this invagination over here, kept going, and buried itself basically and opened up over here on this side. Now here we have an organism that has an opening over here and an opening over here, through and through. Now, if you can just imagine this, if you were to just elongate this a little bit, stretch it out a little bit, this would be the basic structure of a worm. This would be one end of the worm, and that would be the other end of the worm. The head might be over here, and its tail might be over here, and food would just go right through here, through its gut. Now, one of these openings will serve as the mouth, and the other opening will serve as the anus, and this will be the basic structure of the living thing. Now, this blastopore over here is very important because it determines which part of the body is going to be the front end and the back end. If the blastopore develops into the mouth of the living thing, these type of living things, they're called protostomes. Protostomes. Again, if the blastopore goes on to develop into the mouth of the living thing, these type of organisms are called protostomes. And if the blastopore goes on to develop into the anus of the living thing, these type of organisms are called deuterostomes. I remember this as thinking that pro is like number 1. So if the mouth develops from the first opening, they're called protostomes. And duo, as in 2, the mouth develops from the second opening. So if the mouth develops from the first opening, they're called protostomes. This is the first opening. And if the mouth develops from the second opening right here, these are called deuterostomes. In deuterostome, therefore, the blastopore will become the anus. Now humans, believe it or not, are deuterostomes. In other words, we form our bottom first. Okay, the last thing I wanted to suggest to you is the following. The mesoderm is the last of the 3 layers of tissues to develop, and it can develop in 3 different ways. Now the first way it can develop is like this, and the second way it can develop is like this, and the third way it can develop is like this. Now these pictures are cross sections, whereas over here we were looking from on the top, so to speak. Now if the mesoderm obliterates this blastocele, the space that used to be over here, so there's nothing left. In other words, there's no space around here anywhere. This scenario is described over here. So there's no space between the ectoderm, mesoderm, and the endoderm over here. The second thing it can do is it can go kind of hug the ectoderm, and then there's the fluid-filled cavity between the mesoderm and the endoderm. Now this is not the ideal scenario because remember, the endoderm is the gastrointestinal tract, so the nutrients might kind of like flow over there to the rest of the body of the living thing like this. In this case, there is a fluid-filled cavity between the mesoderm and the endoderm. And the third scenario here is the best. The mesoderm kind of hugs both the ectoderm. As you can see, it kind of covers all the way across over here. And then it also hugs the endoderm over here like this. So it covers both the ectoderm and the endoderm, but there's a fluid-filled cavity in between like this, between the mesoderm itself. Now that's very important because this fluid-filled cavity allows for organs to develop inside, to grow inside of the organism. Now in the first scenario here, there's no such cavity, which kind of limits the growth of the internal organs. Here the problem is it's difficult for nutrients to get across. It might be easier to develop organs, but it's difficult for nutrients to get across. Now in the third scenario, the nutrients can get across from here in blood vessels, etc., from the inside layers to all the layers of the body. Needless to say, we belong in the third category over here. Now a cavity is called a coelom. So organisms that do not have such a cavity are called acoelomates or without coeloms. And those that do, like over here, are called coelomates. So organisms that have this fluid-filled cavity, which is very advantageous for the growth of internal organs of the living thing, are called coelomates. And those that do not have it, they're called acoelomates. And this in-between status, where they do have a cavity, but it's not the ideal scenario, these are called pseudo-coelomates. Pseudo meaning false. This diagram, for example, illustrates 3 types of worms. They might look very similar, but in fact they're very different. An earthworm, which is the third one over here, is actually quite a complex living thing, with a circulatory system and everything. Whereas a roundworm, compared to an earthworm, is a very simple organism. Now a flatworm, the first one over here, as you can see, is an acoelomate. It has an ectoderm layer, a mesoderm layer, and an endoderm layer, and there's no cavity in between. A roundworm is a pseudocoelomate. It has an ectoderm layer, and a mesoderm layer, and an endoderm layer, and there's a cavity between the mesoderm and the endoderm layer. Not the ideal scenario. Whereas a coelomate, which is earthworm over here, is quite complex. It has an ectoderm, and it has a mesoderm that both hugs the ectoderm and the endoderm over here. This is the ideal scenario, great for digestion and absorption of nutrients and distribution of the nutrients, and also allowing internal organs to grow within the space thus created. In summary then, what we studied today is early embryonic development, and has suggested that even though the animals in the animal kingdoms are vastly different, their early embryonic development is very similar. They all go through these similar stages. They start out as zygote, and they develop into an embryo, which develops into a blastula. Then one part of it dimples in, or invaginates like this, creating this blastopore over here, which becomes either the mouth or the anus of that living thing. And that really is the beginning of the intestinal tract or the gut. Some living things only have 2 layers, like over here, but more advanced animals have 3 layers. The mesoderm layer usually derives from the endoderm layer like this. In primitive animals, there's only one opening, which serves as both mouth and the anus. But in more advanced animals, blastopore continues and goes straight through a crossover here like this, creating a second opening like that. Now, which one of these openings serves as the mouth and the anus determines if the animal is going to be classified as a protostome or a deuterostome. And then I suggested that the middle layer, which is the last layer to develop, can do one of 3 things. It can obliterate the space, and these are called acoelomates. It can maintain the space, and evolutionarily speaking, this has great advantages. But without the mesoderm layer covering the endoderm layer, and these are pseudocoelomates, and finally the coelomates. The mesoderm layer both hugs the ectoderm layer and the endoderm layer. Okay, now you might be saying, well, how does all of this help me understand this? Allow me to illustrate. Mammalian and human embryology is more complicated because they also have to make the placenta, etc. But at some stage, they do have these 3 layers, the ectoderm, mesoderm, and endoderm layer. What I'm going to do is I'm going to draw this part of this figure over here. So here it is. This is the mesoderm layer, and this over here is the endoderm layer. Now understand that the mesoderm layer has a cavity like this, which I've skipped drawing over here. And outside of this, of course, would be the ectoderm layer like this. But I've skipped those to simplify our diagram. You also understand that this is the primitive gut, this here being the mouth. And you also understand that this is a tubular structure which we're looking in cross-section. It's kind of like a yellow cylinder, and outside the yellow cylinder, a red cylinder. So what we have here is the primitive gut with the endoderm and the mesoderm. So if you can imagine, like over here, the endoderm kind of like pouches out like this. And the mesoderm, of course, just follows like that. So here we have the endoderm pouching like this, and the mesoderm just following along like this. And this endoderm outgrowth is just keep going to grow and divide and branch out like this. And the mesoderm is just going to kind of like, grow along with it and spatialize like that. And on this side, the same thing. The endoderm is just going to keep growing like this. And the mesoderm is just going to follow along like that and specialize. See, if you wanted to, you can call this the pancreas and you can call this the liver. This, of course, is a gross simplification, but you can see, even with a simple understanding of the basic embryological development, how the complex organ systems make sense. I hope you enjoyed. Until next time, as-salatu was-salamu ala rasulillah walhamdulillah rabbil alameen. Assalamu alaikum.